So hopefully you have isolated enough plasmid DNA. So then how do you quantify it? So you will use a spectrophotometer for this step. So it is well known that your DNA molecules can observe UV light. So when your DNA molecules, which is double-stranded conformation, is dissolved in maybe water or TE buffer, they observe a certain amount of UV light. So the spectrophotometer can detect how much of light is observed by your DNA that's dissolved in a liquid sample. Okay, so if we compare that against the blank, okay, which would not have any DNA, just maybe water only, you'll see the difference between your sample and the water, and the difference will give you a clue on how much DNA is dissolved within your sample. Okay, so normally about 50 microgram per mil concentration of double-strand DNA can observe about 1 OD unit. OD is the optical density, that's the observance unit of UV light, which is 260 nanometer range. Okay, so if the concentration is less, of course it's going to observe less UV light. It's going to go down. So let's say it's observing only 0.5 OD, then that means your DNA is less concentrated, about half-fold, which will be about 25 microgram per mil. So you can use that as your reference point. Okay, so the sample that you made on your own, which should have double-strand plasmid DNA, will be measured using the spectrophotometer that's set at 260 nanometer UV light. Okay, but at the end of the last step, you only had like 50 microliter of TE, which dissolved the DNA, right? So you only have like 50 microliter of DNA made from your mini prep plasmid DNA isolation process. And the thing is, for your spectrophotometer measurements, you have to have at least 1 milliliter volume of your sample to be able to use the spectrophotometer. So you cannot, do the measurement with 50 microliter, right? So to come up with enough volume that can be measured on the spectrophotometer, you have to dilute your sample, maybe 1 to 100 dilution. So 1 to 100 dilution, if you're targeting for 1000 microliter, is what? Maybe if you take about 10 microliter of your DNA sample out of 50 total, and then mix that with 990 microliter of water, the pure water that we have in the lab, nano pure water. So it's 10 over 990 plus 10, so it's 10 over 1000, which is what? 10 over 1000 is, if you simplify it, 1 over 100, so there's 1 to 100 dilution, right? So that's going to be how your sample will be diluted. Okay, so you also need 1 mil, 1000 microliter of nano pure water as well, because that's going to be used as a blank. Okay, that doesn't have any DNA. It's going to calibrate your spectrophotometer to read as a zero observance. Okay, so you need that as a reference. Okay, so of course, when you do the spectrophotometer measurements, you need to have your sample inside of a special container called the cuvette. Okay, especially cuvette that's made out of pure glass or quartz is what you're going to need. So transfer your water and diluted DNA into a cuvette. You will be given with those, and then do the measurement at 260 nanometer. Okay, to understand the concentration, as well as at 280 nanometer as well, because 260 nanometer UV light is well absorbed by DNA, okay, RNA, but 280 is well absorbed by protein. So by comparing the concentration, comparing the observance, you can not only estimate the concentration of DNA, you can also estimate how pure your protein, your DNA is. So we'll talk about that. But anyway, if you get a certain reading on the 260 nanometer UV light, okay, so that reading will be multiplied by this reference point, which is 50 microgram per mil. So let's say 260 nanometer reading you got was 0.1, for example, 0.1 OD, then 0.1 times 50 microgram per mil is the concentration. Okay, so which will be what? Okay, 5 microgram per milliliter. Okay, so that'll be the concentration. However, you just made 1 to 100 dilution, right? So you have to consider that dilution factor. So the original sample that is undiluted should be 100 times more concentrated. So you have to multiply 100 to that. So instead of 5 microgram per milliliter, you will get 500 microgram per milliliter. Okay, so that'll be the concentration of your undiluted sample. Okay, makes sense? And then so since you did the 280 observance reading, okay, you can do the observance reading at 260 divided by observance you got from 280 nanometer UV light. Okay, so if you do the ratio 260 observance over 280 observance, okay, that ratio value would give you some idea about how pure your DNA sample is. Let's say the ratio happens to be 1.8. The OD 260 reading was 1.8 times more than 280 reading. That means you have a DNA sample that's mostly pure DNA, double-strand form. Okay, but if that ratio happens to be close to 2, then that means you have a lot of RNA sample in your DNA prep. Okay, if the ratio happens to be lower than 1.8, it's much lower than 1.8, then that means you have a lot of protein mixed up with there. So the ratio of 260 over 280 observance will give you an idea about how pure your DNA sample is. Okay. Okay, finally, your review questions. So I want you to answer these questions. What are plasmid DNA and how are they different from chromosomal DNA? So plasmid DNA are much smaller. They carry maybe non-essential genes, but some important genes like antibiotic-resistant genes in them. Okay, and then what is the main application of plasmid DNA in the modern biotechnology? Of course, they are used as what? Delivery vehicle, vector of your gene of interest into the cell, because they will shuttle back and forth from one cell to another, and then once they are delivered into the cell, they'll be self-replicating, staying in there, and then the gene that they are carrying will be transcribed and translated. So that's a good way of delivering your gene into a host cell. And then what are the main steps in the plasmid DNA mini preparation procedure? Explain what each step does. Okay, number 1, isolating cells from the medium, right? Number 2 is, getting out of medium and mix up the T buffer to weaken the membrane and cell wall structure. Number 3 is lysing the cell membrane cell wall by adding that denaturing solution. Number 3 is neutralizing and then precipitating the heavy membrane debris and plasmid DNA by adding a lot of salt. And then centrifugation, removing those aggregated garbage and getting the DNA out. And then phenylchlorophyll precipitation, purifying your DNA further by removing a lot of proteins. And the ethanol precipitation, bring down your DNA into the bottom of the cell. And washing, getting rid of the salt and SDS. And then drying is to recover your DNA from ethanol. And the resource spending with TE will give you the final plasmid DNA. Okay. What were the concentration and the purity of your plasmid DNA? It's based on your UV reading. 260 will give you the concentration, and the purity is the 260 over 280 will give you the idea about how pure your DNA sample is. Okay, so that's it for this lab.